Hello, everyone. This is Mr. Friedhoff. We're going to be going over the next section or the next chapter in our units on evolution and classification. And this is basically, we're going to be looking at how species are identified or how species are classified using certain scientific names. And with that being said, let's just jump right into it. This is just going to be the area of taxonomy. This is where we classify species, we name them, we show them how they are related to other species. Basically, this is just us are scientists looking at how do we identify and name a species. Now, we don't use common names because the common names that we give just living things can be confusing. For instance, like tomato or coyote, it sounds pretty like, okay, we all know what those things are, but there are many types of tomatoes and there are many types of coyotes. So how do we differentiate these names and make sure we don't have any confusion with these names? So, give you an example, the electric eel really isn't even an eel; it's a fish. The koala bear is not a bear; it's a marsupial. The flying fox is not a flying fox because those don't exist, but it's a bat. The mountain chicken is not a chicken on a mountain; those don't exist. These are frogs. So these common names that we use maybe in everyday language aren't what we use in science. Now, we didn't have a way of really universally classifying species until Carlos Linnaeus came around in the 1700s. He was a Swedish scientist and basically the father of taxonomy. But basically, he wrote a book and kind of showed us how we were going to later figure out, hey, this is the best way to do it, classify species. And this way of classifying species is called the Linnaean classification system. Now, he originally didn't have all of these, but he had most of them. And what he did was he started grouping organisms into more and more specific groups. We have nowadays domain, kingdom, phylum, class, order, family, genus, and species, where domain is the least specific and has the most organisms, and species is the most specific and only has one type of organism. So you can see here, as we go down the list, it becomes more and more specific. We go from domain to species. This is kind of like looking at a like an address or like the location of somebody where their country is going to be the least specific. A lot of people live in the country, but only one person's going to maybe be in a room because as we go down this list from country to state, county, city, neighborhood, street, house, and room, this is just getting more and more specific, just like in that classification system that Linnaeus developed. Now, binomial nomenclature is part of this system. It's called it's the two-name naming system, and this is where we use the genus and species to identify an organism. So you can see here genus and species. The genus is capitalized while the species is not, and they're both italicized. Now, when we make the species name for these species, we usually use Latin, which is a dead language. And the reason why we use Latin is because it doesn't change. The words don't change, the meanings don't change. What one thing means today, it's going to be the same thing tomorrow and the next year and the next year and the next year. So looking at those species I showed you before, you can see their species names is a lot different than their common names. So we might still refer to these things as with their common name, but in science we'll use specifically their species name. Next, we're going to cover the three domains of life, and these are the largest classifications of all of life. And the three domains are bacteria, archaea, and eukarya. Basically, in this section, I want you to be able to identify an organism into one of these three groups. And these are the characteristics of this group of bacteria. They're prokaryotic, which means they don't have a nucleus. They're unicellular, which means they're single-celled. And they're microscopic. They're very small. Now, these organisms can either be heterotrophs or autotrophs. Heterotrophs, remember, mean that they eat the other things for food, and autotrophs mean they make their own food. So they can either be one of those two, and there's thousands of different types of bacteria out there. Now, the one interesting thing that really delineates bacteria from archaea is the substance of something called peptidoglycan. You can see these are the peptidoglycan molecules that we're going to be kind of focusing on for bacteria, but basically these are the signature molecule that really defines bacteria. Because if we look at archaea, they are very similar. You can see they're prokaryotic, they're unicellular, they're microscopic. Most live in extreme environments. So these archaea are very similar to bacteria. However, two things here is their cell walls don't contain peptidoglycan, and they really live in weird environments, a lot weirder than the bacteria do. The last group that we're going to look at is the domain of eukarya. And this is the most diverse group, and it's the group that we belong to. Now, Within the eukarya domain, there are animals, plants, protists, and fungi. And you can see each has more specifics that make them in that kingdom group. Now, obviously, we're animals because we're multicellular and we're heterotropous. We eat other things for food. But you can see each of these kingdoms is just a little bit different. You can see as we look at this tree, we can see how our evolution is a little bit different than what we maybe pictured, where you may have thought bacteria and archaea are more closely related. However, Archaea and Eukarya are the most closely related between the two, or between the three, I should say. But bacteria evolved first, then Archaea and Eukarya very closely second. And you can see as Eukarya evolved, we have these four kingdoms. We are most closely related to fungi, and then we are both most closely related to plants then, and then further back are protists. All right, we're going to get into the third section here, which is going to be about mapping out evolution. We're going to start building things called cladograms. Now, cladograms are diagrams that just basically show how life on Earth is related. So this on the right-hand side is a cladogram. You can see what we have here on the top are species, and then what happens is we show what evolves along the way. So you can see here, jaws are the first thing to evolve. In this case, lampreys don't have jaws, so they evolved first, and they kind of that split before the jaws evolved. The sea bass, this fish, has jaws, but it doesn't have lungs. So we put it kind of splitting off next. Antelopes have jaws and lungs, however, they don't have gizzards, so antelopes then split off, and you can see here bald eagles and alligators both have lizards. Sometimes, again, you'll see another set of like adaptations here or even here along the way, but basically these cladograms allow us to show evolutionary history. And those branching points that I showed you, they are known as speciation events. They're specifically more known as nodes, but these are just speciation events where one species becomes two. And that splitting event shows that species really diverging and kind of going in two different directions. So when we see these speciation events or these nodes, they're just kind of like a population becoming two different species. Our next concept that we're going to kind of tackle here with mapping out evolution is something called ancestral versus derived traits. Ancestral traits are when a characteristic is common because it's inherited from a common ancestor. A derived trait is a newer characteristic that evolves along the way. There's a lot of words there. Let's try to break this down. Ancestral traits are going to be less useful for us to understand how things evolved. Ancestral traits, think like ancestry. This means that they are there because of a common ancestor, and usually all of the species in a cladogram will have them. For instance, like all mammals have fur. That's something that all mammals have. However, not all mammals have like opposable thumbs, or not all mammals are bipedal. That means they walk on 2 feet. So like having opposable thumbs or walking on 2 feet is a derived trait for like humans, where the derived trait evolved later on. And this helps us really identify what is in a certain group and what isn't in a certain group. So these traits are going to be a little bit different with how we look at them. And when we look at certain cladogram charts, we're going to be looking at how can we make an evolutionary tree based on these charts. Now, you can see here the ancestral trait here is A, where all of the species have them. But like B, C, D, and E are derived traits because they evolved along the way and they, again, help us map out the evolutionary tree just a little bit more specifically because they evolved later on. Now, these characteristics can be many different things. They can be like traits, behaviors, DNA. They can be a lot of different things that we can organize an evolutionary tree based on. But this is basically what a cladogram is. So I do want to go over how to use one of these tables to make a cladogram. So you can see what the X means is that that species has that adaptation. So the first adaptation that we're going to draw here, I'm going to draw my line. The first adaptation that we're going to draw here are the legs. Now we're not actually going to draw the legs. What we're going to do is we're going to draw this adaptation being right here. Okay, so I'm going to label all my adaptations in red, and I'm going to label this one legs, because all of these organisms have legs. So again, the adaptations are going to go kind of next to these lines, and then the only organism that has legs is the lizard. So again, I'm going to bring this down because there was a speciation event, and that's going to go right here. Okay, the next adaptation to evolve is fur because rabbits and fur, or rabbits have fur and legs. So I'm going to make a little kind of adaptation line there. I'm going to put fur. I'm going to bring this one a little bit closer so they know it's right there. I'm also going to bring lizard down just a little bit. There we go. And then what I'm going to do is I'm going to have rabbits speciate from that. There's going to be a speciation event. I'm going to put rabbits, rabbits, and rabbits are going to go here. So again, rabbits have legs and they also have fur. Now, dogs are going to speciate next. Again, we're going to put that adaptation, and they are a carnivore. So I'm going to put carnivore. I'm going to put carnivore on top, or actually on the side here, because that is a adaptation. And then lastly, I'm going to put dog on the last one there. All right. This is how you make a cladogram. So you can see these X's represent that species having a certain trait. So legs, all of them have legs. Rabbits and dogs have fur, and dogs are the only carnivores. So again, rabbits have legs and fur, but they're not a carnivore. Lizards don't have fur and are not a carnivore because they just have legs. So that's how you work through a cladogram. All right, the last section here is going to be, again, on mapping out the tree of life. And basically, we're going to be looking at what clades are first. And a clade is a group of organisms that come from a common ancestor. This includes both the living and extinct species. So you can see here on this cladogram, or this tree of life, this phylogenetic tree, whatever you want to call it, this is not a clade and this is not a clade because it's not one common ancestor in all of its descendants. These two on this side are clades because you can see here, this is one common ancestor and all of its descendants. One common ancestor and all of its descendants. So this is what a clade is. It's a group of organisms and all of its descendants. You might think, well, why do we need to know this? Well, when we try to group organisms or name organisms, we usually typically give a name for a group of organisms that's based on one common ancestor they have and all of its descendants. For instance, like dinosaurs. We want to know what the first dinosaur was, and then we call everything after that a dinosaur. That's why birds are dinosaurs, is because they are an ancestor, or they have an ancestor with the common ancestor of that dinosaur. So again, this helps us kind of group things a little bit differently in science and in biology to help us understand what is something and what is not something. One of the things that we have to understand about these cladograms or clades is that the nodes can rotate. These speciation events can rotate. So these trees here, they are all the same. Even though we have rotated some of these speciation events, they are still the same tree. If you look here, this triangle and this rectangle are still the closest two together. On each of these trees, that triangle and rectangle are the closest two species that are related. If you look at the next step, the star is the next one in line. And if you look at each one of these trees, that star is still the next in line. Then it goes square and circle. But even though they're in different locations, these still show the same tree. So there are multiple right answers when we make these phylogenetic trees and these cladograms. Now, just to show you a little bit more about these trees and what they show us is the common ancestor of two species is located at that speciation event. So you can see between B and C, this is the most recent common ancestor. Between A, B, and C, the most recent common ancestor is here. So these common ancestors really show us how related we are to other species. And we can track those speciation events, or we can track where those common ancestors may have lived based on these things called phylogenetic trees. So what I can show you here is like taxon 1 and 2 share a common ancestor at A. Taxon 1 and let's say taxon 4, we have to go all the way back here to D because that's the most recent common ancestor that both of them had. Between like taxon 8 and taxon 10, the most recent common ancestor is I. Between like taxon 6 and taxon 13, again, we'll go back in time and find that ancestor H is the most common ancestor between that, again, 6 and 13. Now, one last thing we have to go over is something called a phylogenetic tree, and this is basically a cladogram that just shows time. So you can see the cladogram, there is no time on this chart, but in a phylogenetic tree, these speciation events are specific. They are important to show when certain aspects of the speciation events occurred. So this is a phylogenetic tree because we can see on the bottom here, there's a time scale. So like this shows us when speciation events may have occurred. It's like humans and mice, our most recent common ancestors about 65 million years ago. Us and chickens, we have to go back all the way to here. And it's about 300 million years ago. But basically, again, these nodes or these speciation events show us specific common ancestors, but based on time. So again, like zebrafish and frog, if we go back and we show that most recent common ancestor, it's probably about 350 million years ago. Now, here's another example of a phylogenetic tree. You can see again, here's our lineage. We can see that present day only D, E, and I are still living. All the other ones have gone extinct, like C, F, G, H, B, A, and J. These are all extinct species. So we can still look at common ancestry. So, like if we look at what's the most recent common ancestor between D and C, that's going to be H, because this is the first point that both of them will diverge from. The most recent common ancestor between like I and D is going to be like G, because we go back in time and we find the first location where both of D and I are most closely related to, and that's G. All of these species are related to A then, because A is the first species that began this evolutionary tree.